Hello students, welcome to BioSmart Academy. In today's lecture, we will continue our lecture series on enzyme, and this will be the third lecture on enzyme, that is how enzyme works. Okay, how enzyme work. So I hope that you enjoy the previous two lecture, that is lecture 1 and 2 on enzymes. Okay, so students, please subscribe and share if you like the lectures, and share to all the students, because I make all these videos free of cost, and I want that each and every student get all these lectures. Even you can watch all the lectures that were previously uploaded in my YouTube channel. Okay, so let's start today's lecture. So first of all, what should we going to study today? So today we were going to study about enzyme, that is how it work. Okay, what are the some of the important site is there in enzyme where basically substrate bind and complex is forms. All this we will going to study today. We will also going to study about the binding energy. Okay, so all these things we will going to study in today's lecture. So let's start today's lecture, that is how enzymes work. Okay, see, first of all, just look at this figure. Okay, so if you look at this figure, okay, and from this figure I explain all the things. I will going to explain all the things. Okay, so please note down all the things. Okay, so in our previous lectures we discussed that enzyme work by lowering the activation energy, by lowering the activation energy. That is, if activation energy, by lowering the activation energy, that is activation energy we can represent as EA. So enzyme works by lowering the activation energy. So when the activation energy become lower, then rate of reaction increase, then rate of reaction become increase. Okay, and how enzyme lower the activation energy? Enzyme lower the activation energy, air enzyme lower the activation energy through binding energy, okay, that is the binding energy basically helps in lower the activation energy. So binding energy basically helps in lowering the activation energy, binding energy. Clear? So basically we can write binding energy as del G and B. This is a symbol of binding energy. Okay, so basically we read all these things in our lecture 1. Okay, so now I hope that you get these things clear. Okay, so now in today's lecture we will discuss in details how does, or you can say from where this binding energy basically come, from where this binding energy basically arise. Okay, from today's lecture we will understand all the concepts, all the mechanisms of binding energy. Okay, so let's start today's lecture. See, if you look at this figure, okay, so this is an basically structure of an enzyme. This is a structure of an enzyme. Okay, that is chymotrypsin. Clear? So this is chymotrypsin enzyme. Okay, so if you look at this enzyme, so here a active site is present. This is the active site. Okay, here you can see the label is there, that is key active site amino acid residue. So what does this active site means? Okay, so basically just note it down. Active site basically is a pocket of an enzyme, where the reaction takes place. Okay, or you can say that enzyme catalyzed reaction takes place within the confines pockets of enzyme, which is called active site. Okay, so this is a definition of active site, or more simply you can say that active site is a region of enzyme where substrate bind, where substrate bind this is a substrate, where substrate bind and catalysis occur. So this is a definition of active site of an enzyme. Clear? Now I hope that you understand the proper meaning of active site. Okay, now so when a substrate basically bind to this active site of enzyme, then enzyme plus substrate combine and it gives rise to a enzyme substrate complex. Okay, I will show you with the help of an equation. Okay, now so what does this substrate means? What do you mean by substrate? Okay, so here the substrate means that the molecule that is bound in the active site and acted upon by enzyme. Okay, the molecule that is bound in the active site of an enzyme. The molecule that is bound in the active site of an enzyme and it is acted upon by enzyme. It is called substrate. Okay, now remember, so this is the chymotrypsin enzyme, and remember one point, each and every enzyme is substrate specific. Okay, so that is the unique physical and chemical properties of the active site of an enzyme. If you see, this is the active site of an enzyme, and if this is the active site of an enzyme, and this active site is basically substrate specific. Okay, this is substrate specific. Only a specific type of substrate can bind to the specific type of active site of an enzyme. Okay, now a question is arise, why a specific type of substrate is bind to a specific type of enzyme? The answer is that due to the arrangement of atoms in the active site. Here you can see that amino acid residues. So a substrate is made up of different arrangement of atoms. Active site is basically you can say made up of different arrangement of atoms. Okay, and the arrangement of atoms make the active site of an specific for specific substrate. So if a question is arise that why the active site of an enzyme is substrate specific, so the answer will be the specificity or the active site of an enzyme is substrate specific due to the arrangement of atoms. Okay, due to the arrangement of atoms. Clear? So if we see active site, suppose I just write down here, suppose if you see the active site, okay, if you see the active site, then the surface of the active site, the surface of the active site is lined with amino acid residues, clear, amino acid residues with a subsequent group, and all these amino acid residues have some subsequent groups, and within this group a substrate is basically bind, and the enzyme catalysis takes place. That's why the active site of an enzyme is basically substrate specific. Okay, so now I hope you get the concept clear why the active site is substrate specific. Okay, so now again just look at the enzyme. If you again look at the enzyme, that this is the active site of an enzyme. Okay, this is the active site of an enzyme. Here basically substrate is bind, and the substrate basically the active site is substrate specific. Okay, till now you get all these ideas. Okay, now another question is arise that how the enzyme is basically bind with the substrate. Okay, there is need of something, because whenever there is some interaction is there, it needs some types of force. Okay, it needs some types of force. Suppose A is want to combine with B, then it needs some force through which they combine. So basically here in enzyme, in case of enzyme, the substrate bind to the active site. Okay, this substrate is going to bind to this active site of an enzyme. Okay, the substrate is going to bind to this active site of enzyme by multiple weak non-covalent interaction. Okay, by multiple weak non-covalent interactions. Remember this. Okay, non-covalent interaction. Okay, so basically substrate bind to the active site of enzyme through non-covalent interactions. Clear? And remember one thing is that in our biological system, most of the interaction is non-covalent type. Okay, clear? And what are these interactions? These interactions are hydrophobic interaction, hydrogen bonding, ionic interaction, all these types of interactions are basically present. Okay, I already take a class on all these type of interaction. If you see in our playlist of biochemistry, the first lecture is of about the interactions. Okay, now, so whenever a interaction is occur, whenever there is an interaction, you always remember a free energy is released. Okay, a free energy is released. Suppose A is basically going to interact with B. A is basically going to interact with B to form AB. Okay, so whenever a interaction is takes place, so there is some energy release. There is some free energy released. There is some free energy release, and same thing happen in this case when substrate basically bind to the active site of enzyme through non-covalent interaction. A free energy is released. A free energy is released. Clear? Free energy is released. Okay, and this free energy that is released through the interaction of the enzyme and the substrate, we call this free energy as binding energy. Okay, binding energy. Clear? Binding energy. Okay, clear? Now just look at here. Suppose this is an enzyme. Suppose enzyme plus substrate, it gives rise to enzyme plus substrate complex. Okay, and this enzyme and substrate are combined through non-covalent interaction. Okay, non-covalent interaction. Clear? Non-covalent interaction. And whenever there is a type of interaction, whenever there is an interaction, some amount of energy is released, and here that energy is known as free energy, and that free energy is known as binding energy. Is known as binding energy. Binding energy. And this binding energy can be written as del GB. Okay, binding energy. Okay, so in this way binding energy is released. I hope now you get my point. So basically if I define binding energy, so what will be the definition of binding energy? Just note it down. Binding energy is the energy that is derived from enzyme substrate interaction. From enzyme substrate interaction. So it is a form of energy that is derived from enzyme substrate interaction. Or more specifically, you can say binding energy is a major source of free energy. As I already told you, binding energy is a major source of free energy used by enzyme to lower the activation energy, that is EA. Clear? So binding energy is the major source of free energy that is utilized by enzyme to lower the activation energy, so that rate of reaction can be increased. Rate of reaction can be increased. Okay, so this is all about the binding energy. Okay, now students, I hope you get the concepts clear till now. Okay, now, so till now you get all the things correct. Okay, now there is another one thing is that we learn about active site, we learn about substrate, we learn about specificity of this substrate, why it needed. Okay, we learn about binding energy. Okay, now another one thing is that the specificity of enzyme substrate interaction that we basically discussed earlier is arises mainly from hydrogen bonding and shape of the active site. Remember this. Okay, it means that it needs some complementary shapes. Okay, some complementary is required. Okay, and based on this basically a model is described that is known as lock and key model. Now, so what is lock and key model? Okay, so if you look at this figure, okay, so this is basically an enzyme. Okay, and the enzyme basically have active site which is complementary, that is exact complementary of this for the substrate. Okay, so basically this is the unbound state of the enzyme. Okay, and here this is a bound state. This is a bound state of the enzyme, bound state of the enzyme after the substrate basically bound. So in this figure, you notice that this figure represents lock and key model, lock and key model, lock and key model. Clear? So what does lock and key model explain? The lock and key model explain high degree of complementarity, high degree of complementarity between the shape of substrate, between the shape of substrate and the enzyme active site, and the enzyme's active site. Okay, so it explained the complementarity, and this model was given by Emil Fischer. This model was given by Emil Fischer. Okay, now if I draw a figure, then you will understand more. Suppose this is an enzyme. Suppose this is an enzyme. Okay, this enzyme, and the substrate is made in such a way that it basically going to bind with like this. If I draw with different color. Okay, so substrate is basically going to bind just complementary to each other. So this is a substrate, exact complementary. Okay, so this is the lock and key model. Okay, so this model explain the complementarity between the shape of the substrate and the geometry of the binding site of enzyme, or you can say the active site of an enzyme. So this is lock and key model. But students, remember one thing, it is very rare that you find exact complementary. Okay, it is very rare. Okay, and another one thing is that exact complementary is also not good for a proper, or you can say active enzyme catalysis. Okay, now why it is so? Because now if you look at this graph, okay, so here three graphs are there. In basically case 1, here three cases are there. In case 1, there is no enzyme. Okay, so substrate is a metallistic. Okay, substrate is a metallistic, then substrate is going to form transition state when the metallistic basically bent, that is bent stick, and the product will form when the stick is basically broken. So basically in the first case there is no enzyme, so the reaction takes place as usual, and in this case you can see the transition state is so high. Okay, because there is no enzyme, but a transition state is formed. Okay, I will take a separate lecture on transition state of enzyme. Okay, so now coming back to this topic. Okay, so this is in the first case there is no enzyme. Okay, so substrate basically going to form transition state, and the transition state then going to form product. It means here we can see that transition state is necessary for the formation of product. Okay, now if you look at the second case, enzyme is present. Okay, and this enzyme is complementary, is exact complementary to this substrate. You can see here exact complementary to this substrate. So here enzyme substrate complex is formed, and due to the exact complementary of the substrate unable to bend. Here you can find a missing state here, and that state is known as transition state. So in this case transition state is not formed. So what does this mean? This means that enzyme complementary to the substrate, but here the transition state is not formed because the complementary caused the enzyme so rigid that it unable to bend, that is unable to going to form transition state, and due to that a few product is formed, and so we can say it will not going to form active product. Okay, so this is the second case. Now if you see the third case, this is a third case. Here enzyme is complementary to the transition state. Okay, enzyme is complementary to the transition state. So here basically enzyme substrate complex is formed. Here the enzyme is bind with the substrate, no problem, but it is not complementary to the substrate. Okay, here the second stage is formed, that is the transition state, and in transition state enzyme is complementary to each transition state, and when you can find enzyme is complementary to the transition state, then transition state is formed, and it gives rise to a product, a active products. Okay, so here the interaction is more powerful as compared to other. Okay, and the binding energy that basically release, as I already told you, when enzyme substrate basically lowers the activation energy. So transition state is here. What does this means? Okay, so basically when a particular enzyme reached a transition state, then in that case maximal binding energy is released. So the maximal binding energy released when the enzyme facilitate the formation of transition state. Remember this, note it down. Maximal binding energy is released when an enzyme facilitates the formation of this transition state, and this is only possible only when the enzyme is complementary to the transition state. Complementary to the transition state. Okay, so what is transition state? Here the transition state is a point of highest free energy, where the free energy is highest, in which reactant, that is substrate, is partially converted into product, not fully, partially, and after that it will give rise to active products. Okay, so here this is the concept. So students, I hope that you like today's lecture, and if you like today's lecture, please hit the like button, subscribe and share with all other students, because I want that everyone get all these free lectures. This will going to benefit you for your NET examination, okay, and all other entrance examination. Okay, now in our next lecture we will going to discuss in details about this transition state mechanism. We will going to explain the graph of the transition state, that what happen when enzyme is present, when enzyme is absent, when enzyme is present, what is the effect of enzyme on the transition states. We will discuss in details in our next lecture about the transition state. Okay, so I hope you find today's lecture is very helpful. Here we learn many things. Okay, so thank you. We will meet on our next lecture. Till then, thank you.